Hello students, in today's session let's learn about flow cytometry, one of the most advanced imaging techniques with a great deal of applications in biology. Flow cytometry is an analytical technique that's used to analyze the physical and chemical characteristics of particles in a fluid as it passes through a laser beam. The cell components are fluorescently labeled and then injected into the flow cytometer. The fluorescent particles are excited by a laser to emit light at varying wavelengths. Before we go into the details of flow cytometry, let's understand a little bit about the history behind this technique. Flow cytometry was introduced in the year 1968 by Wolfgang Gode, a professor at the University of Münster in Germany. It was later commercialized by the company called Partec. Now Partec has been acquired by Sysmex and called Sysmex Partec. At the time of his invention, flow cytometry was called pulse cytometry. 10 years later, in 1978, at the conference of the American Engineering Foundation in Pensacola, Florida, the technique's name was changed to flow cytometry, the term that quickly became popular. Soon after its discovery, flow cytometry instruments were developed and released into the market, such as cytofluorograph, which came out in 1971 from Biophysics Systems Incorporated. Another popular flow cytometry instrument was ICP-22, which was released by Partec in 1975. The first fluorescence assorted cell sorting, or FACS in short, instrument was used to sort cells based on the principle of flow cytometry, was made and sold by Becton Dickinson in 1974. Currently, FACS and flow cytometry equipment is supplied by major vendors such as Becton Dickinson, Sysmex and Agilent Systems. Some of the most popular models currently in use are BD FACS-Verse from Becton Dickinson, ACA NovaCyte from Agilent and Sysmex Cytoflow Cube from Sysmex Partec. Now that we have seen the history and the models of flow cytometers, let's go into the principle of flow cytometry. In flow cytometry, the fluorescence can be measured to determine various properties of single particles, such as cells. Up to thousands of particles per second can be analyzed as they pass through the liquid stream within the flow cell of the flow cytometer. The properties of the fluorescent particles measured include particles' relative granularity, size and fluorescence intensity, as well as its internal complexity. An optical to electronic coupling system is used to record the way in which the particles emit fluorescence and scatters the incident light from the laser. Flow cytometer instrument has three main components. They are the fluidics, the optics and the electronics. The term flow cytometry is based on the fluidic flow of particles passing through a quartz chamber, also called a flow cuvette. Particles from the sample are pumped through a flow cuvette in a very narrow stream following inside a larger sheet stream of water or saline, which is used to focus the sample stream into the center. Any cell or particle that's 0.2 to 150 micrometers in size can be analyzed. If the cells are from a solid tissue, they are required to be disaggregated before they are analyzed. Also, the cells from animals, plants, bacteria, yeast or algae are usually measured. Other particles such as chromosomes or nuclei can also be examined. Some particles such as marine algae are usually fluorescent, but in general fluorescent labels, which are fluorescent chemicals, are required to tag the components of the particles. For example, cells are stained with propydium iodide, which in turn fluoresces the DNA contained within them. The section of the fluid stream that contains particles is referred to as the sample core. The optic system is made up of lasers which illuminate the particles present in the stream as they pass through and scatter light from the laser. Fluorescent molecules that are on the particle emit fluorescence, which is detected by carefully positioned lenses. Generally, the light scattered from up to 6 or more different fluorescences is determined for two different angles. Optical filters and beam splitters then direct the light signals to the relevant detectors, which emit electronic signals proportional to the signals that hit them. Data can then be collected on each particle or event, and the characteristics of those events or particles are determined based on the fluorescence and light scattering properties. The electronic system is used to change the light signals detected by the optical system into electronic pulses that can be detected by a computer. The data can then be studied to ascertain information about large number of cells over a short period of time. Information on the heterogeneity and different subsets within cell populations can be identified and measured. Some instruments have sorting feature in the electronic system that can be used to change to charge and deflect particles so that certain cell populations can be sorted for further analysis. The data are usually presented in the form of a single parameter histograms or as plots of correlated parameters, which are referred to as cytograms. Cytograms may display data in the form of a dot plot, contour plot or a density plot. After going through the brief working of the flow cytometer, now let's discuss about the applications of this powerful method. Flow cytometry is used to perform several procedures including cell counting, cell sorting, detection of biomarkers and protein engineering. Flow cytometry has numerous applications in science, including those relevant to healthcare. The technology has been widely used in the diagnosis of health conditions, particularly diseases of the blood such as leukemia. Although it is commonly used in various fields in clinical practice, it is an important tool in basic biological research as well as clinical trials. Some examples of the fields this technology is used include molecular biology, immunology, pathology, marine science and plant biology. In cell biology, flow cytometry is primarily used to detect the live cells from dead cells with proper fluorescent tags that differentiate them. Another important biological application is its use in accurately detecting the amount of DNA within the nucleus, which in turn determines its ploidy level. In medicine, flow cytometry is a vital laboratory process used in transplantation, oncology, hematology, genetics and prenatal diagnostics. In marine biology, the abundance and distribution of photosynthetic phytoplankton can be analyzed. Flow cytometry can also be used in the field of protein engineering to help identify cell surface protein variants. With this, we come to the end of this video on flow cytometry. Hope you have got a good insight into this method. Until I meet you again with another analytical technique, bye.